Hello and welcome. This is Bhaskar Napte from Pharma Growth Hub. And as a part of today's video, we are going to understand what is meant by OOS, OOT, and OOE, and how these three terms are different from each other. So let us begin with the presentation. And let us first understand what is meant by out of specification result. And here is the definition. A result that falls outside established acceptance criteria which have been established in official compendia and or by company documentation such as specification. We all know that each and every product, be it excipient, inactive raw material, active raw material, finished product, has to come along with certain number of tests and its predefined acceptance criteria or the limits. For example, if you have a limit for assay of a drug substance like paracetamol as a not less than 99% and not more than 101%. So that becomes the specification for paracetamol drug substance. And during the analysis, maybe at the release time, you have found that okay, for some reason the assay result is not within the 99 to 101%, then you can say now I have come across a out of specification result for assay. And just this is an example for your understanding. The limit for assay is 95 to 105%, and this is a drug product, right? For drug product, this kind of limits are very much accepted, and the reported assay value is 92.20%. So I was speaking about the paracetamol drug substance example before that, and this is another example for your understanding. I hope you understand what is meant by out of specification result. And out of specification result is not only for chemical analysis, it can be about physical, chemical, or biological analysis too. It can happen for the physical parameters of tablets like hardness, like friability, like the appearance of the tablet. So anything which is not meeting your predefined specification, limit, or acceptance criteria, you can say now it is the out of specification result. And it is applicable for routine testing during the releasing of the batch. It is also applicable for the in-process testing if you have some in-process parameter as a part of your process control, and if something goes out of the limit, then for in-process stage the out of specification procedure also applicable, and also it is applicable for the stability study too. I hope you understand what is meant by out of specification and where does it really applicable. It is also applicable for the vendor source raw materials. Suppose if you are getting some raw materials from the X vendor and if you are testing that raw material into your lab to understand its compliance, and if something goes wrong during the testing, I mean if the result is not meeting the specification, again you have to investigate the failure through OOS investigation procedure. Clear? Let us understand now the out of expectation result. Out of expectation, the OOE, an analytical, an atypical, aberrant, or anomalous result within a series of results obtained over a short period of time is an OOE result. Atypical means what? Which is not really expected by you. Aberrant means okay, something was not yet observed for the another batches or another product. And anomalous like suspected that we are not very much sure about those results. Within a series of results, within a series means what? Suppose you are conducting a dissolution test for a drug product. Dissolution test for a drug product, and as a part of your testing procedure, you supposed to execute the dissolution for 6 units, right? Now this series of results means what? You have 6 different results for the dissolution of that particular batch. So out of this series of 6 observations, if one or two observations are not in line with the another observations and they are different from the rest of the observation, then you can say now this one or two results are a typical result. or they are anomalous result, they are aberrant, or they are out of expectation. So you conduct the dissolution only for that particular time period, right? Maybe for 30 minutes, maybe for 24 hours, according to your drug release procedure. So it's just a for short period of time, only for that particular dissolution. It is not over a period of 1 month or 6 months. You are compiling the dissolution data and then trying to plot that. That is not the case. It is only for very short period of time, only limited to that particular analysis or the sequence of analysis. Very importantly, an OOE result is a result that meets specification. It must meet the specification. Otherwise, if the result is outside the specification, we just discussed the OOS, the out of specification term. So the moment the result goes out of the specification, the OOT is no more applicable. You have to investigate the failure with the out of specification procedure. So remember one thing, the OOE result may be atypical, may be aberrant, may be anomalous, not as expected by you, but still it meets the specification. Still, it meets this specification. It's a very important statement. But for some reason, it is outside the expected variability by the analytical procedure. And very importantly, it is observed for over a short period of time. Over a short period of time. And I will explain why I am reiterating this term short period of time when we discuss about the OOT term. That is the uh, you know, uh, out of trend term, right? OOT means out of trend. So for this time, let us understand that the O is something which is a typical, aberrant, anomalous, not as expected by you, but for a short period of time. But for a short period of time, and all the results are still meeting the specification. Let me explain you with the help of example now. And here is the case of a drug product for a dissolution parameter, and the limit is how much? Not less than 75%. Now look at the sample number 1 or the unit number 1. It is 86% drug release. Sample number 2 got 91%. Sample number 3 got 97%. Sample number 4 got 131%. But let me know whether this is out of the specification. What is the specification for dissolution? It is not less than 75. Not less than 75. So is it less than 75? Absolutely not. So I can say that it meets the specification for dissolution. But if you look at the another values like 1 unit, 2 unit, 3 unit, 5 and 6th unit, these all values, right, statistically very close to each other. But when you compare the sample number 4, it is it is not as expected by you. And the dissolution is performed for a short period of time. You are not performing sample number 1 today, sample number 2 tomorrow, and sample number 3 day after, and sample number 4 in the next week, right? You are conducting the dissolution run by using the same dissolution apparatus, by dropping all the 6 units at the same time, collecting the sample at same time, and analyzing by using the same instrument, maybe HPLC. So the short period of time requirement has been met within a series of result. So are there a series of result found? Am I talking about only single result? Absolutely not. There are 6 results. Yes or no? And out of this series of 6 result, which is obtained over a period of short time, I got sample number 4 out of expectation. And hence I must investigate this failure or observation. I may not say failure, but the observation by out of expectation SOP. I hope you got the meaning of out of expectation. Remember that it is still meeting the specification. Let us now talk about the third term, that is the out of trend or OOT result. What is meant by that? A time dependent result which falls outside prediction interval or fails a statistical process control criteria. Time dependent term is very important. There is a set of result observed right? And it is collected over a period of long time. If you remember, while discussing about OOE, that is out of expectation, we said that okay, now these are the series of results observed in a short period of time. But in case of OOT, when the results are gathered over a period of long time, let us say, for example, stability study. You say, I have a 12th month stability result. Then you will compare that 12th month result against initial, then 3 month, then 6 month, and then ninth month, and finally the 12th month. So you are talking about not a result over a short period of time, like within the same day, but you are actually comparing the result over a very long period of time, over a 12th month period of time. And for some reason, if the results are not within your prediction window, like trend, that you are expecting some trend to be followed for impurities, for dissolution, for assay, but for some reason, the result for assay or result for dissolution or result for impurity for X time point, maybe ninth month time point, is not following the trend, maybe decreasing in the assay values, maybe increase in the impurity values. So this is your predefined trend based on to the earlier time point. And if that trend is not followed by the current time point, which is let us say 12th month time point, then you can say, okay, now I have come across out of a trend result for X parameter. I hope you understand the difference between OOT and OE. And remember, even in case of OOT, the result is meeting the specification. OOT result is a result that meets the specification. It is just outside the trend. Otherwise, if the result gets out of the specification, you cannot investigate that particular observation by OOT, but you have to investigate this observation by out of specification, the OOS. So for both OOT and OOE, your result must meet the specification. Your result must meet the specification. So what is the OOE means? Let me brief you one more time. OOE means the series of results, observed over a short period of time. And if some results are outside your expectation, okay, but they are still meeting the specification, then okay, you will say, I will apply the OOE SOP. Second part, if the results are observed for a long period of time, the way I explained the stability result, but the results are meeting the specification, but some results are not within the trend. They are not following the exact trend. And then you can say, now I come across a out of trend result, and you have to investigate those observations by OOT SOP. I hope you are now clear on OOS, OOE, and OOT. I'm sure you must be. And this is an example of OOT result. The stability study of the drug product, the assay limit is 90 to 110%, initial value is 99, third month 99.1, sixth month 98.5, and ninth month suddenly you got 108.8. Now remember, this 108.8% result, is it meeting the spec? Absolutely yes, because my specification limit is 90 to 110%. So the result is within the 90 to 110%. But is the ninth month result following the trend? Absolutely not. It is something out of the trend. And hence I must apply the OOT, out of trend SOP, to investigate this particular observation. And why not OOE? Because this observation is not over a short period of time. This is a not observation done over the same day. It has been acquired for over a ninth month of time, initial month, then third, sixth, ninth. So the OOT is applicable overshare. I hope you now got the better clarity about what is meant by OS, what is meant by OE, and what is meant by OOT. Thank you so much.